So I don't know, how am I doing for time? Okay. I want to move more into the diagnostic component of the meeting. As I said, in between were a number of talks that detected XMRV and didn't detect XMRV. So I'm really not going to spend time defending the people who believe they're right and defending the people who believe they're wrong. I hope over the next year we'll understand, we'll meet in the middle and realize there may be some very small practical issues that would not be foremost in our minds as we went into this, but are certainly foremost in our mind now. So these talks I'm not going to present. This is work that's ongoing at the National Cancer Institute up in Frederick with Mary Kearney's group, developing two assays, XSCA, which is a single copy assay, ultra-sensitive method of XMRV detection, and XSGS, a single genome sequencing assay for sequencing large genome fragments. In other words, this allows you to look at each individual DNA. And so both were prepared in Mary's lab. Both are actually up and running. And I think I changed my presentation. I hope there's one more. There's one more piece of information coming down here that you don't have. But the results, and I'm going to talk about this panel again. This was work done and presented by Graham Simmons. Using a blinded panel of spiked patient samples, they can detect the XSCA method detects XMRV with very high sensitivity, down to almost a single copy per milliliter of blood or plasma. XMRV and mouse endogenous MLVs are very highly genetically related, and it's important to make a clear distinction between them when we are examining the XMRV prevalence. And between them, the XSCA and XSGS. For HIV, the SCA assay is the gold standard. Our goal here is to develop the equivalent assay for XMRV. But between XSCA and XSGS, we can discriminate between XMRV and endogenous murine leukemia viruses with 100% accuracy. And as I said, what's ongoing at the moment with Mary's group, we have 2 prostate cancer studies at the NCI. Each have 100 patients. We have a MAX study with approximately 1,600 patients. There's a study ongoing in patients who have prostate cancer and HIV. This is a very small number. And Mary's group will also be working with the blood working group, panels 2 and 3. I'll talk about that in a few minutes. Serodiagnosis. There were a couple of presentations here, one from the Abbott group and a second from the NCI. I'm just summarizing the work from the Abbott group, the serodiagnosis, the reagents. These are recombinant proteins expressed in E. coli, or mammalian expression for the surface proteins, the surface glycoproteins. We have multiple assay formats. We have a number of positive and negative samples. But I want to make another point with the next slide, developing serological reagents. Well, this is just to show. Here I'm going back to the rhesus macaque experiment shortly after infection and showing the various protein antibodies to the various proteins coming up on day 9. I'm not sure if you can see it from your end, but at about day 9 after infection, we see the surface glycoprotein, GP70. Day 11, P15 protein comes up, and at day 14, P30 comes up. And this is using an XMRV lysate, and then using the serum from the monkeys. But this is a point I wanted to stop for a minute, because recently we had a reporter from the Wall Street Journal at Frederick. I thought it was very important to explain how and why and how much time it takes us to get from A to B when we're developing either serological reagents or nucleic acid detection reagents. This is a slide from the protein expression group at NCI Frederick, who have cloned and expressed, purified every XMRV protein. As I said, the fortunate thing is it's a very simple retrovirus. Here we have the structural proteins, matrix, P12 capsid, and nucleic capsid, the enzymes, the protease, the reverse transcriptase, the integrase, and the membrane proteins. So now we've successfully cloned, expressed, and purified 8 XMRV proteins. They're going to be the basis of a serological diagnosis. Which ones do we use? Which ones do we use in combination? how many do we have to use? So how many proteins does a patient's serum have to be positive to be defined as positive? How many does it have to react to be defined as positive? And so now basically we have 1, 2, 3, 4, 5, 9 proteins where we go into this sort of cookbook chemistry of mixing and matching to define the best reagents for serological diagnosis. And this is what takes time. This is the easy part of our work. This is recombinant DNA technology, which you can probably teach at middle school now, never mind high school. This can be done in days. The next part, to developing an assay and in a format for high throughput and knowing which are the best of these viral antigens to use, this is where we slow down. We can't make this go faster, and this is why it takes several months. And we understand for a CFS patient tomorrow is too long. But this is why in our case it takes a while to go from all the reagents we have to which are the ones we need. The same report from Rachel Bagney actually started looking at some of the samples from the Low and Alter study. One of the criticisms of the Low and Alter study was that there was no serology done. This was all PCR based. And actually Rachel is detecting, this is the envelope glycoprotein. When nucleic acid was detected, Rachel can actually see antibodies to the surface glycoprotein. This is a very limited study at the moment. One of the other things that we'd like to do as we're developing, again, a thread that came through the meeting is that there is no standalone assay for the prevalence of XMRV. There was one study from a lab in Texas that was only nucleic acid based, and the point was made quite strongly that we cannot diagnose and stand on XMRV detection based on a single assay. The goal is to develop nucleic acid detection, serological detection, and this is a third assay that we've developed at the National Cancer Institute, which is virus replication, which is probably the best detection method. If you have replicating virus, then you can be pretty sure you've got XMRV there. The problem was the original tests that were set up were rather time-consuming and really could not be done on a scale, a multi-sample scale. These would take several weeks. Well, again, with a little bit of retroviral trickery, a cell line was developed called the DERSI cell line. DERSI are the detectors of exogenous retroviral sequence elements. As one of our former colleagues at the NCI who developed this cell line. And basically a gene is split in two and is non-functional until after XMRV infection. As I said, by a little bit of retroviral trickery, the two halves of the gene come together. The gene that they actually they lead to, is a gene that encodes a fluorescent protein. And now you see that 3 days after infection of these cells with XMRV, we can detect virus replication, these little green blobs. I hope you can see them. This is XMRV infected cells. And so we've successfully taken these methods from several weeks down to several days. I should point out this is not a front-line diagnostic. We will not be doing, if we need to test several thousand samples, we will not be doing virus culture as a front line. This is designed as a backup assay to complement the nucleic acid and serological detection methods. The other thing we've tried to do very hard while we've been preparing our reagents in the last year is to disseminate them amongst the public. I've been a beneficiary of the AIDS reagent repository for almost 2 decades. This was set up 20 years ago, where researchers are allowed to donate their reagents to the repository for use by the research community. We felt it was very important to do the same for XMRV, to let researchers have access to the materials we've been preparing at the National Cancer Institute. So far at the DNA level, we've put about 64 recombinant clones into the repository. The next line is to hopefully deposit antibodies and cell lines for expressing monoclonal antibodies. But again, I think it's very important that as we move through our intramural program, that we make as much of this available. Although we're developing serological nucleic acid and virus detection assays, we are not demanding that those are the assays people use. We can make our reagents available to others for use either as diagnostic or for basic research purposes.